Good afternoon. Thank you for coming to the Institute for Quantitative Biomedicine seminar series. I'm biased because I invited him, but this is the talk I'm most excited for this semester. But don't miss our final two talks, Hassan Honavar from Penn State and Stephen Hacker from Leiden. So let me give a brief introduction to Wen while Mike puts the slides over. So Wen and I trained in the same lab many years apart, part of the Enrico Fermi lineage. And by the time I was a young student, Wen's contributions to methods in cryo-EM were so fundamental that my thesis completely relied on them. So going back further in Wen's life, he did his undergraduate degree at Peking University, which, if you have no familiarity with universities in the People's Republic of China, is kind of like as Peking University is to China as Harvard is to the US, both in terms of prestige, but also in a deep history. He went on to do his master's degree at the Chinese Academy of Sciences, and then he came to Baylor College of Medicine, which was at that time a real locus of EM methods development, where he did his doctoral training and his postdoctoral training with Professor Hua Chu at what was then the National Center for Macromolecular Imaging, which was a P41 funded center for methods development in cryo-EM, where Wen worked on some of the fundamental breakthroughs that led later to the resolution revolution in terms of, for example, in computational methods, understanding CTF, understanding how particles come together, et cetera, and wrote his own package even for single particle analysis called JSPR or Jasper. I'm not sure which way you pronounce it. But Wen went on to Purdue, where in addition to his continued work on methods development and solving a number of interesting structures, he also directed the cryo-EM facility there and was the principal investigator of a large U24 consortium award supporting investigators from many different academic institutions around the region, including, I believe, Professor Diane, Professor Kulchik from QB, in doing their international quality data collection on the instruments under Wen's supervision at Purdue. So Wen has recently moved to be more of a neighbor to us at Penn State, not too near, but not too far, as part of the national reshuffling of cryo-EM people from one place to another, where Wen holds the Huck Endowed Chair in Structural Biology and is the director of the big cryo-EM facility over there. So in terms of research, I will point out first that, in my opinion as a scientist, a lot of the major breakthroughs that we're able to make in biology came because there were experiments that previously were impossible, and someone made a methods advancement that made it possible. Wen has been on the forefront of methods development, and that has led him into a number of interesting applications. That was largely viruses, many years ago, including asymmetric reconstructions. And now, as Wen and others render more difficult macromolecular complexes tractable, has led him into neurodegeneration and amyloids. So I'm grateful that you drove out to see us, and thank you for being here, and we look forward to your talk today. Thank you.
Thank you, Jason, for such a wonderful introduction. So I'm honored to come to the place where PDB is. So I have been all worshipped and the leadership role of PDB in the structural biology. I'm glad I have the opportunity to be here. So let me, okay, yeah. Yeah, so So as Jason has mentioned that I actually started as the physicist background, then entered in cryo-EM field, most focused on methodology aspect. So as part of that effort, we're also using applied cryo-EM to some biological systems. So today we're going to focus on one type for the system in recent years is on the structure of protein amyloids that involved in neurodegenerative diseases. So as a part of this, I will first describe some of the structures we solved using cryo-EM. Then we will follow up with some of the technical challenges that we face during that process and how we develop a new method to help us to solve that. So this is just a very brief introduction my lab. So my lab is fundamentally a cryo-EM focused centered lab. We care about both the method and applications, but our heart is in the cryo-EM method aspect. So we look at the biological problems or challenging at the time that in the current method couldn't solve. We'll be very excited to develop a new method to address those. So throughout the years, so we have, this effort led us to address many areas of cryo-EM challenges, including the sample preparations. For example, we developed affinity grid approaches. We also spent a lot of time on the image analysis from single particles, and then more recently in helical processing. So today, we're going to using two aspects of this a very broad research interest to illustrate some aspect of recent research. So this shows some of the cryo-EM structures throughout the years that we determined either as a pure application project or as a benchmark sample for technical developments. So we largely grouped them into a different category. So in the very early days, cryo-EM, so our application most focused on the aggregation viruses. The reason was that in early days, the very large in size and high symmetry really made the viruses much more friendly for high resolution studies. So later, and after this breakthrough in detectors, then the success in high resolution cryo-EM structural viruses has then extended to less symmetric objects. So as part of that work, and we also realized that many viruses do not just have this symmetric exahedral capsid share, but also have some non-exahedral components. For example, bacteriophages have this unique tail vertex. Right. Then also some of these double-stranded RNA viruses have this built-in RNA polymerases inside. So there's a very fascinating assembly. So we developed a computation method to allow us to determine the entire structure, not just the symmetric shares, as shown by this very first basically breakthrough at that time to see the porter protein with 12-fold copies and a 6-fold tail complex at this porter tail vertex. So actually, this was a more recent work that actually we had actually a chance to collaborate with Jason on this particular sample just 2 or 3 years ago, published together. So then in recent years, as the technology expanded beyond viruses, as part of this cryo-EM, effort, especially in my facility role and technical development role, we have been expanding the application to this other protein complexes. For example, so we have this is the very first non-virus structure we studied in my lab on the polymerase, probably roughly about 10 years ago. Then recent years, we have collaborations on metabolic enzyme in acetylcholine complexes. So and then we have the P97 protein complex as part of affinity grid approach. And we have another cancer-related protein complex, PMD5, with a small ligand development in this area. We just submitted this paper the other day. So, but the actual focus, the application area is in this neurodegenerative diseases area, where we study a lot of the protein amyloids structures like tau, prion, A beta, and I will come back to this very interesting new discovery later. So today we're going to first go through some of the the structure aspect of this amyloid structures and focus on the diversity of the amyloids. Before going on to details, just a very brief introduction of the protein neurodegenerative diseases and the role of the protein amyloids in those diseases. So we all know that Alzheimer is very kind of serious disease, especially as the population is aging. So there are a lot of people being affected by this Alzheimer. For example, the 6 million in the US suffering from this, mostly from the elderly. So when young AD patient, the brain will suffer damage compared to the healthy brain. It's shrunk and a lot of deformations. So the medical or cell biology study has pinpointed that this one of this highly correlated damages to the brain is correlated to the protein tau filaments, as shown here. First image using EM about 40 years ago in '85. So current understanding of the role of the amyloids, the tau amyloids, is that the tau is a microtubule binding protein that a normal function, it is a bind microtubule and stabilize microtubule, for example, in the long exons to so then when in the pathological conditions, the tau will be hyperphosphorylated, then you lose the binding capability, then will fall off from microtubule, but microtubule become destabilized, cause brain neuron damages. At the same time, this highly phosphorylated tau would self-aggregate into these tangles accumulated inside the neuron cells and cause follow-up damage. So this correlation is very strong, although the exact molecular mechanism how this amyloid accumulation and the brain damage is actually occurring is actually still require a lot of details studies. So our role in this using cryo-EM is to characterize this tau and other amyloid structures from different patients suffering different, say, neurodegenerative diseases, including AD and other amyloidosis. So one thing is immediately you can see that this is amyloids tend to aggregate. So not only inside the brain cells, but also in the extracted sample. That actually turned out one of the real challenge in cryo-EM studies. So because we require dispersed particles, right, for single particle imaging analysis. So that actually turned out one of the real recurring, probably #1 challenge in our studies. I will come back to this later part of this talk. So so this actually goes beyond AD. The other neurodegenerative diseases, right? So starting from many years ago, more than a century years ago, it's already being known from medical field that many different diseases, including Alzheimer, Parkinson, and others. Then there are clinical subtypes, all right? So then in more recent studies, right, so we associated those diseases to different proteins that form these amyloid fibrils. And of course, for diagnosis for other early screenings, right, the field developed, identified multiple biomarkers. So from this diagram, it will show that, this pretty complex association of different neurodegenerative diseases, different subtypes of these diseases, and different amyloid proteins. Okay, so this is quite well known. But from a structure aspect, right, how we actually understand these details, there's a connection between these diseases and the structure level that is only available after the cryo-EM revolution occurred in recent years. So the general understanding of this is a amyloid related to how it's related to neurodegenerative diseases is roughly in this very abstract diagram. So the proteins, whether it's tau, A beta, or prion, first exist in say soluble monomers. Somehow then began to form oligomers, first benign, then maybe become these toxic oligomers further aggregates and form this long fibers, and then become mature and then causes it down. This is the abstract concept. This is kind of a working hypothesis in the field. So then the details still to be worked out. I will come back to the challenge is that we actually do not know how each protein under different biological or chemical conditions, what type of filament you will form. Actually, that become a very serious gap, still remaining gap nowadays. So all these amyloid fibers have a common structure features is this cross beta sheet. So it roughly shown in this diagram. So this is one of the representative EM images of one type of amyloid fibers. So if one would zoom in into this, one would see this general features is that basically each subunit of the protein will form one layer of this the beta sheet structures, and the different subunit will stack up along a long axis, form a helical twisting across beta structure again. So how do we know this general features? Because using X-ray fiber diffraction, not a crystal, it's just fiber diffraction, just like this famous double-stranded DNA photo 51 is same technique. Fiber diffraction, one would see very prominent features around 4.7 angstrom that correspond to spacing of neighbor the beta strands. So then the little more faint around 6 to 10 angstrom is basically distance roughly between this beta strand and the neighbor beta strand in the same layer. Okay, so this was the best state-of-the-art structure information for many years until cryo-EM became so powerful, could determine atomic model resolution starting 2017 by shared group. So my lab was approached by our collaborator, Professor Ruben Vidal and Dino Gedi at IU School of Medicine. So we set up a collaboration, study the cryo-EM structure of amyloids extracted from patient brains. So those patients unfortunately suffered from these diseases and passed away. So they donated the brains for the research. So throughout, we started roughly around 5 or 6 years ago. Then throughout the years, we solved many this amyloid structure, which I will show you later. But as a basic guide for the next few slides, I would roughly divide this into these 4 sections, all centered around this central concept called structural diversity of protein amyloids. So there's 4 different aspects of diversity. So starting with, so there's more than one protein that can form amyloids. Then in the same patient brain sample, there are more than one type protein amyloid, either from the same protein, but a different fold or packings, or just different proteins. And that's more seriously is that there's, from the same protein, they can fold differently and in different diseases. And then they are distinct across different diseases of same protein, but the same fold for the different patients of the same type of diseases. So this has actually become one of the real puzzling and challenge nowadays in the field. Okay. So the tightly correlated to this is that when we try to recapitulate the patient form this amyloid in test tube or in the cell or mouse models, we failed to produce the same protein amyloid structure that as in the patient form, we still struggle how to do that. Okay, I will show you more details in later. So we started with this first diversity is that the more than one type of protein that can form amyloids. So is this? There's not only this intrinsic disordered protein, it's well known that are forming the amyloids, but some surprises in recent years. Some well-folded protein in normal conditions. Nobody would suspect they would form amyloids, but then we found that actually they can form amyloid under certain conditions. So this is just a small gallery of the amyloid structures we have studied and solved in the last few years. So we have many more currently being in the lab before we publish. So it's not listed here. So you can see that, the many different proteins, starting from the most well-known tau proteins, and then a few years we solved the prion amyloids. Then, they have solved the A beta and either in different type of diseases, the all involving A beta. And then we also some less known is called the Danish family diseases, ADAN. So you see the many different proteins can form amyloids, but these are actually well-known cases. It's many years ago, we already know that they form amyloids. So the real surprise, in our study was a few years ago. So we studied a system that we found this protein, TMAM, is a membrane protein in 106B. In one of the patient sample received and saw a very good structure. We found that surprise finding that is a new protein. Okay, so we'll go through some of the details of that. Serendipitous discovery of a new protein. Okay, so the story was this. So one of this patient sample, our collaborator sent it to us. We imaged. So this actually give you a sense what kind of ex vivo patient brain extract samples look like. Immediately you can see, right, the filaments. Those are good, what we want. But at the same time, it's a lot of the contaminants. Basically it's far from the ideal sample we normally enjoy from a recombinant pure proteins. So this is actually one of the challenge, but this challenge in relative sense is solvable. It's suboptimal, but it's okay. So my student Raja solved the structure quickly. So we saw two type of amyloids fiber in this data set. So one type is you see this lot of the fudgy edges, right? So this actually correspond to the tau protein in a particular fold we call AGD fold. So that's already previously seen in other tauopathies, not in this, but we say this tau can form the same fold in different tauopathies. So, but what really bothered us was the other amyloid structure actually solved at a much better quality than the tau proteins. So, and very clear backbone and side chain, and with a very large, just fuzzy density, probably correspond to the modifications of the amino acid. So we suspect there are probably glycosylation sites because they're very large, much larger than the other modifications. So initially we thought, okay, so because from a pathology from others, so this should be another form of tau. We began to place the atomic model into this, which could never get the satisfactory validation stats. Just do not really fit well to the density maps. Then this triggered us to look at maybe this is a different protein, unknown proteins. That time a few years ago, we have no idea how to identify a protein purely from a EM density map, but that was a real challenge to us. So we tried two approaches. First, we look at the large modification densities spaced across certain distances, right? Because by counting because we can see the backbone very well, we can roughly estimate the spacing, how many amino acid part they are in between the neighbor modifications. We'll assume this is a glycosylation site. We will build up a pattern using ProSite to search, right? So that is very specific to that. We allowed us to identify some candidates. But what really exciting was that later we realized that most of it actually borrowed from the method pioneered by Professor Hong Zhou at that time, together with, I think, the Finnish group, to determine they call the bio-ID approach. Is that from the density map, then each of the amino acids self position, this local shape of the voxel value distributions. Then you try to guess what type of amino acid each of the this C alpha region should correspond to. Due to the resolution, that estimate is very, basically fuzzy. Okay, so you can only give a rough probability for each one of the 20. So each one would be very poor quality. But when you have the sequence, say 20, 30, then that information become a very rich information. Then you can use that as a profile using, say, hidden Markov chain sequence search to search against the entire protein sequence that we know, right? So then this is actually the approach, a more general approach that in the end we chose. So this was using the Phoenix program implemented at time to rank against all these human proteins in against this density map. Then we found that there's one clear top ranked match, which is a TMM1R6B that nobody ever knew that was associated with neurodegenerative diseases or forming amyloids. At the same time, the tau protein, the tau has multiple isoforms. So we will basically highlight the tau, the score for tau, this is hidden in the background. So it's not a tau at all. So, and this is the e-value, this is so, especially local e-value, is so good. So essentially, you find this, you just trust it. And it's more than 10 orders better than second hit, right? So then we'll look at, so we're building the model using this protein sequence and a very good validation stats. So what is really intriguing was that, so if you look at it in the literature, what this protein is a lysosomal protein, transmembrane domain, and this domain that form an amyloid correspond to the lumen region of this protein. So inside and if you just using the structure prediction method that time before AlphaFold time, it has very good confidence prediction that is from this very well folded structure. Not like this, the N-terminal is very, right? So AlphaFold will fold this similarly as well. So this is actually not intrinsic disordered region at all. So, but it's now we found it's actually from very well-ordered amyloid structures. So this is a very intriguing to us at the time. So, so during the similar time, the other two groups also independently identified this. So the question for us, for pretty much everybody, is that in the native source sample, right? So how many other amyloids we might have mixed? Okay, in the old days, we did not have this capability. Now we have this, but does the extraction process or our image analysis process allow us to get everything, or we're not missing anything? Every time we look at a new sample, a new data set, we still keep this in mind. Have we explained everything in our data? Yeah. So one thing we do in the lab is we are very methodology focused group. So whenever we have a new technique aspect, we try to make this more available. So instead of relying on a little bit tricky, very complex process, we build a web app so allow this process to be much more streamlined. Yeah.
Briefly, so when you were looking at AlphaFold predicting that structure that you showed, did it also predict your amyloid?
No, it doesn't predict the amyloid. It predicts something like that. This is actually exactly this amyloid,
If
the
you
alpha
treat it
prediction.
as
Yeah,
a multimer somehow,
yeah.
like how it would be possibly AlphaFold 3 could.
Yeah, so later, so we have not revisited this. If this AlphaFold 3 or after the database has been many more amyloid, especially for this protein, so AlphaFold might, trained with those information, update might be different. Yeah, but that time, this was not known, not many amyloids, basically no structure prediction would predict world amyloid structures. Yeah.
They rely on the protein interactions
Yes.
within the fibril.
Yeah.
Which AlphaFold 3 might be able to address.
Yes. Yeah. So as part of that, so we actually, my student and we basically streamlined this analysis process into accessible web app. So added this address. So you just provide a density map and basically self positions. You will go through all this process and present a very nice the ranking plot for you, and also see. So what are the trustable and with enough of good e-values by default is better than 10^-3. So the rest will be just the wrong match. So so then so the next aspect of structural diversity is that the multiple protein amyloids exist in the same brain samples. So I will just show you this example here. This is still unpublished, but I will show you a very nice example. So this is the cryo-EM images of this sample, right? So you immediately see, say for example, this is very different, distinct from this. Then my postdoc, Xueyong has gone through very detailed analysis, actually struggled for a while to fully, probably less than fully, resolved this amyloid structure in this data set. So this just give you a glimpse of what this amyloids look like in 2D class averages. So you can see the different type. So more than 10 type of unique amyloid structures. So Xueyong could solve 9 out of this, but still the 2 actually still very hard to solve, still not solved. This is give you basically evidence, actual case that solving amyloid structure is actually very tricky. So some cases like tau, most time is pretty straightforward, but A beta and some others actually can be very tricky. Yeah. So this is just show you this little bit the atomic model placed in the 3D density map. So what you have seen, for the amyloid structure, see, we're not seeing the typical, camera or Primal 3D, this surface view, right? Mostly we showed like this, basically like 2D images, so this is already surface view, but very, very often we say, for example, we're showing here as a 2D view of this. It's because this amyloid cross beta structure, the real unique protein fold mostly in a single layer, single 2D layer. So very often show in this way. So it's about a 9 out of the single data set. This probably has the most so far among all this amyloid sample. So this 9 include multiple type of amyloids, say A beta 42, and actually A beta 40, the 2 variants of the A beta proteins. And then we have this tau, the 2 type of tau protein amyloids. Then we have this TMEM 106B that I just mentioned. Okay, this is the very first time we have seen in our hand in another patient sample. But what's very interesting in this particular one, we saw a new type of A beta structure. We have so far have not seen any other patient sample yet. Yeah. So that's the then we'll focus on the next actually very major challenge in the amyloid study, actual findings from the cryo-EM studies that so the same protein can exist in distinct fold across different disease causing by the same protein. Okay, for example, tau has a different type of tauopathies. AD is just one type. So, but also intriguingly that if you have multiple patients, doctors diagnose as the same subtype, say tauopathies, right? AD or other tauopathies. Across different patients, the fold actually be the same. Okay, so that's a very tight correlation. Okay, so we don't admire that the doctors have done such a wonderful job just based on the symptoms. Those are basically the classification of disease occurred so well to atomic structures. So this must be some real intrinsic science there. At this moment, we still struggle to truly understand the molecular basis for that. So just to give you one example. For example, we studied this AD with the cotton wool plaque that it means certain shape, unique shape features of this plaques. So we solved this A beta structure. We solved other tau structure as well. I'm not showing here. This just show you that this A beta structures it exists in multiple type of the amyloids and especially the different packings of these amyloids. So we also studied the other disease, Down syndrome sample. It's not a real neurodegenerative disease. So Down syndrome, it is mostly caused by the 3 copies chromosome 21. So, it has a lot of, issues in the early on in the life, but later in the life, it almost always develops some AD-like symptoms. So we studied those brain sample Down syndrome patients. Then we found a lot of this amyloid. But you can see in this particular Down syndrome, we find actually 2 unique A beta fibrils that are not seen in any previous reported neurodegenerative disease. So we named it type 3A and type 3B. Type 3B basically the dimer of the type 3A. So this is a unique one. So later we have some other samples that we found the similar one not published yet. So it just expanded these 2 cases. So as shown, so this is in this red box, we have seen the A beta structure right in Down syndrome in form of this multiple type. And then in the cotton wool plaque form this type. Then we have the A beta in another familiar AD with particular mutation that will form very distinct fold and packing as well. And we have several other studies that are not included here, so we have not published yet. So we have seen even more distinct A beta fold. For A beta, such a short peptide, 42 amino acid or 40 amino acid, it's amazing that it can form so many types. We still have not exhausted the old fold yet. Every time we look at a new patient sample, then we find actually new type A beta structures. So just to give you a bigger picture, so this is one of the review papers by someone's summarize all these A beta structures available at that time. You see, this is just a list. So now, since then, we have many more types to add to this list. So this is diversity, same protein from different fold and different packing, right? It's not unique to A beta alone. So tau, it is very well known as well. Same tau, okay, depends on kind of the isoform of diseases, different tauopathies. Then you form many different unique type the fold. So this is true for this A beta and the tau, and it's also for prion, which we studied a few years back. So this is just a very brief introduction of the prion series and how the prion is actually underlying this very serious diseases, right? So Professor Stanley Prisoner advocated for this prion series where protein as the infectious agent, which is a very novel concept, right? Took a lot of effort to establish that, but then this was well recognized, and Nobel Prize was awarded to Professor Prisoner for this prion series. But after many years, we're still trying to get the structure of the prion from human patients. So our collaborator sent us the samples. My student Kadir actually, took this challenge. As a Euro, we face a lot of challenges. For example, the number one challenge for prion was the very bad aggregation. So we had to go through all the different biochemical treatment, the sonication, right? So to disperse to a sufficient level, it's far from ideal, but it's sufficient enough for us to brute force and collect a lot of images and get enough data to solve it. So we solved the structure. So immediately you find that this in a prion forming many different type of the filaments, right? So then we solved it. So we focus on the two types. We call it type 1 and the type 2. So you see, this is the dimer, basically centered with the helical axis here at the center of the dimer interfaces. Then the other two types is helical axis is so off from this dimers. It's very interesting. Actually, this two type actually, look at its glance, you think it's very similar, right? But it's actually distinct because one is the two protofilaments are parallel, the other is antiparallel. So, but the fold, the subunit fold, they actually it is the same. So then we try to use this atomic model from this patient samples. So then we try to understand the common mutations in this protein, right? So then for this particular protein fold, at least we have two patient cases. One is heterozygous M and V, the other is homozygous V. This actually, the VV homozygous has about 10 years early onset of the symptom than the heterozygous MV patient. So from this structure, we try to rationalize why that could be the case. Just look at where this is the M or the V position in this very tightly, this small hydrophobic pocket here. Then if you place V here, it is enough space there. But if we put M there, it has a serious clashes. So we rationalize that this phenomenon property is only formed by the V variant, not the M variant. When you have a V and a V homozygous, it'd be much easier to form this amyloid. But when you have M heterozygous allele, then this property will, in a sense, serve as a competitive inhibitor of this amyloid formation. So that delayed the onset. So the several other mutation probably could be explained correlated to this structure as well, but we have not finished solving this structure yet. Okay, then we look at this the prion structure we solved. Is this the common or unique structure? Then we look at the landscape that time. Then we found that actually the many model system solved for the prion. You can just glance, you see the structure we solved from a human patient brain is completely different from the other model systems. So this is the only patient form. Okay, so this is just give you another example where this is the same protein from distinct fold depending on the context, the environment. So that is across different patients, but the same phenomena apply to the model system. Then we have distinct fold in the model system, so we cannot recapitulate the patient forms. So I was showing here, right? So I will start with this quote from Richard Feynman, the famous physicist, quantum physicist, right? What I cannot create, I do not understand. Okay, so I think in biochemistry we try to understand life by using test tube, right? So there's a lot of effort trying to grow, say, fibers in test tube, in cell or mouse models. For example, shown here. So this is the dominant model, test tube model for tau is based on a heparin-induced tau filament formation. So this was done many years ago in in 96. So this has since been used as a standard test tube model system for tau. Many papers have been published using this model system. But with the assumption that this tau, heparin tau form filament will be the same as the, say, the tau fibers in AD patients. But after this cryo-EM revolution, I think Shor's lab published this heparin tau structure. He found that this is distinct from patient forms. So now basically it's challenging to understand, so why the same protein forms so many different, why in test tube in the model system we can form a very different ones, right? Or what's the value of the heparin tau model system? What's the conclusion drawn from those model system? We need to revisit all those results now. So just some in abstract aspect, this is kind of a generic understanding of protein. In a typical soluble protein or it's well-folded protein, right? So structural biology, we would actually think that okay, so a protein have one sequence will form a very well-defined global minimum native structure. We normally think about this. But then when you have this amyloid, right, this aggregation unfolding others, so you will form an amyloid in a much deeper energy wear and very, very, very narrow and deep. And once you get there, you can never come back. And that's why we got in trouble. But in this cartoon, you see just showing a single amyloid. But now with this cryo-EM structure, diverse structure, we found that we need to expand this to many different, very sharp wear. But the challenge is that we know there are many, but we don't know how each biochemical condition will drive the photon one or any one of those. So that's the challenge. Okay. So in my lab, we try to to see if we can using test tube synthetic peptide recombinant proteins to be able to reproduce some of the patient formed amyloid. So far, we have failed. So this just give you one example for each protein we tried. We tried the tau in our own hand using heparin, but it's from a distinct fold from the reported heparin tau. Then we have the A-beta and the A-dan. None of them look like the patient form we have solved. So the gap is in the field, not only in my lab, is that so far we rely on the readout is based on very complex, slow cryo-EM imaging and 3D reconstruction. So how can we screen numerous conditions to find the right condition that will give us, say, the AD tau form or other amyloid, right? I guess this is similar to the X-ray crystallography in older days. How do you screen numerous conditions to find the crystals? Now we need a similar high throughput screening method, especially the readout method. How do we know the amyloid? It is actually not only exists in the amyloid, but what type of the amyloids, the typing, unfortunately, we don't have that. Now we're thinking about some spectroscopy method, fluorescence, others. It'd be great if we chat with some of you on this aspect, how we can figure out that method. Okay, so that we can narrow down this large screening space, then only using cryo-EM for the subset, the short list. So that is the biologic aspect structure, but look like I'm a little bit too slow, so I will basically just quickly go through some of the computational challenges that we faced and we developed a method for this purpose. So in some sample and contamination, aggregation, as you see here, okay? But there are a lot of this image analysis challenges. For example, how many types, how do we determine the twist and rise of the helical symmetry, and how to obtain initial model, okay? So I'll just very quickly go through some of this, okay? So this is just the helical structures. It has a unique properties that along the axis, you see all the different views. Single image in series, if long enough, you have all the views you need. You don't need many, okay? So, but the challenge is that, the helical symmetry is not one symmetry, it's a family of an infinite number of combination of twist and rise. So how do we determine the twist and rise? So I will skip that, okay? So one of the things in the helical structure, in the same data sets, we always have multiple types, right? So I already show you that. And you try to understand why. It turns out actually helical structure can be considered starting with a 2D crystal Then you just basically draw, say, from one unit cell to the next unit cell, then you just roll it up and then form the helical structure. But depending on how you pick this tool, okay, for example, here, the exact same 2D lattice, you pick up a different pair of this unit cell, then you form a distinct helices. So now I think this helps us to understand this helical symmetry. This is mostly a learning education tool. We have the web app for that. So then we also, the multiple type, how do we know how many type and separate the particle filament to different types? So actually my student borrowed this large language model behind the ChatGPT, right? A sentence of different words. We treat each of this region segment as a different word. Then we try to use a similar this machine learning method embedding type thing. Then we can cluster them, then separate different types. So then more recently, we have this Helicon software just published a few months ago. So then we started using this we leverage 2D classification results, determine the pitch distances, and which then allows to calculate the twist. Actually, more excitingly, we actually, pose the 3D reconstruction of helical structure as the linear regression problem as shown here. Basic idea is we only need to determine the unit cell as a single asymmetry unit here. But the other part is related by mathematical duplication, right, based on the helical twist and rise. Then we can use that set up as a large linear equations, then solve the X is the 3D initial model. The B is the images. The A is the projection matrices. So we solved. Actually, this demonstrated this quickly showing through this. By doing this, okay, we can actually determine the structure from the single raw filament images as shown here, and also the low twist amyloid. Basically, the images, the class 2D averages, only cover a portion of the whole pitch. So that means the equivalent to missing wedge or preferred orientation problem. So we can actually leverage some of the compositional trick is based on the regularization in the linear regression. So be able to obtain pretty good high quality initial models and then determine the twist. So by using this approach, we actually look at one of this tests in real world is an Empire data set previously published. Then my student could actually identify a new, very small number, very new novel type that not reported, and found is actually dimer of a previous published this as a unit. Now we found a new dimer type. The interesting part is that these are only very few, total of 44 filaments, but we could get it to 4.2 answer. But if we just using the two of the longest one among the 44, we still could get a reasonable good 3D structure. So for helical structure, one of the benefit is that you do not need that many. A few long filament, that's all you need actually to get in a nice structure. So the last one, so is I'm coming to the PDB. I mentioned something related to the database. So as part of this helical reconstruction, so we are challenged, right? First, try to learn from this previous solve. Then, my student and I were looking through and found, it seems that some of the information not quite accurate or robust. For example, shown here, obviously, these amyloid structures, delta Z cannot be 1.175 angstrom. There must be some error there. So, so then we happen to have a very nice tool, and we published a few years ago, okay? That can unwrap a helical structure as a 2D lattice for you to very intuitively understand the twist and the rise, simply just as basically the crystal lattice indexing problem. So we'll bring this helical to the crystallography. So by doing that, we actually have gone through the entire list of helical structure in the EMDB. We found roughly around 14% of the metadata helical twist and rise actually have some issues. For example, not deposited, not provided, or say incorrect values, or just typos, or swapped this two fields, something like that. So I show one of the example for each of the categories. From here, we have a complete list and shared publicly on the GitHub. So one interesting thing is that for helical symmetry, sometimes we could actually using a suboptimal partial symmetry instead of the complete symmetry. Basically, the idea is this: if you so this is the optimum twist and right, right? So if you're using this and the other, they're all correct, just partial, right? Just like say fourfold symmetry, you're only using twofold, something like that. So actually, we found a few cases like that. You know, my student, we took the half maps, applied the full optimum complete symmetry. We could see actually small improvement in the resolution. Some cases with with much significant, but we don't have the raw images, so we are limited by what we can improve. So but hopefully this will bring awareness to some of the database metadata issue if they are correct. I hope that later this deposition process or the validation process could actually fix those issues in the future. So we'll be happy to contribute this method to this effort. So I think with that, I will acknowledge thanks to my students throughout the years who contributed to many of this research for past members and then current members. But today, I mostly mentioned the work by those students, postdocs highlighted in red here. Then I have a lot of collaboration with many groups, but today's amyloid collab from the collaboration with the Professor Ruben Vedil and Dino Getty at the High School of Medicine. So thank the funding from the NIH and all the sources throughout the years. So this is our current lab at the Christmas dinner last year at Penn State. So I will stop here and thank you for your attention.
Thank you very much for that talk. And we'll take questions both from people on the Zoom and in the room. And I note that at peak, we had, besides us people logging on for technical purposes, over 40 people on the Zoom. So good turnout today. Any questions? Who wants to start off?
Thank you for the terrific talk. That was really interesting and it's a lot of impressive work. So you mentioned earlier that different mutations have shown different degrees of aggression for the disease, like the V129 was shown to present an onset 10 years prior. Is the suspicion that in patients, assuming no mutations, just polymorphism of the same sequence, is the suspicion that different morphologies will be more aggressive than other morphologies? I imagine with the difficulty of getting in vitro ex vivo samples, it's probably difficult to study that at this point, but is that the general suspicion?
Yeah, I think this is a very good question. This is probably very deep medical science, probably beyond my expertise. So, but definitely, as I mentioned, right, different disease subtype of the same disease have a distinct the filaments, right? Even just from the same AD, there are many different subtypes. For example, the H2V1 has very unique approach from the standard sporadic or median. So indeed, for the H2V, actually that mutation that there's some report that actually from the it's actually protective mutation because there's a lot. The reality probably is more complex than some mutation might be delayed, some might accelerate, and depending on the other co-pathology, other contact, for example, the Down syndrome. Why it's not just AD itself, it's actually probably compounded by the the duplication of entire chromosome. So, but yeah, I think definitely that is one thing very important to study. In cryo-EM, what we can contribute at this moment is I think we are helping to gather a lot of observations and we'll correlate it to those medical record and pathology diagnosis of other things. Hopefully, by integrating all the data, we can learn a real rule of others. I think right now we are still at this, I think maybe early midpoint of data collection. Before we solve, we really don't know how much data, what type of data we have to solve it. So, but hopefully, we can contribute to that process.
Thank you for the
Sorry. Very nice talk. I have a quick question about recombinant versus like the protein that you actually find in the human tissue, the disparity. Is there any speculation? Is it because recombinant tau protein, for example, doesn't have all the necessary post-translation modifications?
Yeah, so definitely, I think begin to have this picture, right? So indeed, from I think so far, probably the only success in the time from the source group is that. So I think the two effort, one is using just the all repeat region without the other NOC terminals. So that could form, one of this type, PHF type. Then the other, it was, many modifications to mimic these phosphorylations at a certain selected site that probably will neutralize the charge in the state and bring this, the charge interaction to a state compatible for that. So, but then, there's a contact, the cell tissue or cell contact and the contact of, say, intensity of different salt, pH, and other modification. They probably all contributed in the end what form it will fold. So right now, to form a distinct fold is the typical outcome. To form a particular one, nobody really can predict. It's just by chance you form that, we will observe it. Do we know how to change condition with directly to a particular one? We actually do not know at this moment. So hopefully that will be one with this research and effort and outcome in the coming years. We actually really learn that rule. We know what a biochemical condition will drive to a particular one. We have real clear, in a sense, first principle level understanding. Right now we don't.
Okay, I'm gonna jump in with a follow-up question. Then we've got one from Ruben in the chat, and then Eddie will get the last question. So I wanted to follow up. 10 years from now, what I expect we'll be seeing from your group or others is inside the cell, in situ, looking at the structure of these amyloids. Today, you still have to do some fractionation and get appropriate condition for this helical reconstruction. So what I want to ask you is, what are the ways that we might still, even though you've made a big advance going to patient samples, what are the things that still might be going wrong, and what do you see as the key technical and methodological steps in taking out these fibrils that we need to think about when we read papers in this field?
Yeah, definitely. That's what as a technical person in the EM field, I think a lot about that. So our current is called extraction and forcefully disperse the aggregates so that we can use a single particle cryo-EM, right? So then, I was dreaming, okay, but we have not been able to do that, but I was planning to do this. So for example, as shown here, so you see, number one challenge in amyloid study is the aggregation. So some we could disaggregate with reasonable effort, some we just could never, okay? So then I started to think, why do we want to disperse them to use a single particle? Why don't we just using tomography, collect this, and using subatomic averaging of those to solve it as it is in the aggregate state? That's one baby step towards what right? So we're trying that. We have not been able to do that yet. Okay, but ultimately, what I really want is let's just study this aggregates, right, in the brain tissue using the current called focused ion beam sending to get, of course, with a clamp to first to identify where they are, then using fib to cut the other part and only retain the one with the aggregates. Then using tomography and this subatomic average to get a high resolution, the true in situ amyloid plaque in the human brain tissue sections. So that's my ultimate goal. That's actually one of the reasons I really want to have a new opportunity, environment, resources to do that. Maybe that's part of my move. Actually. Thank you.
So a question from the chat. The phosphorylation of tyrosine, serine, and threonine. Could you say a few words about the contribution of that phosphorylation to the types of fibrils and disease pathology, particular for tau? And further along that line, in the high resolution structures you have solved and presented here, can you detect phosphorylation of the side chains?
Yeah, so a lot of the details is around the quality, right? So for exactly the shape is actually very often is not that clean cut. So to, you know, to have to identify phosphorylation or the modification with real certainty, right? So probably really hard. But once you combine it with the sequence with no mutation, we kind of basically say, oh yeah, look like this, for example, this TMM106B, very large, fuzzy, weak density around this asparagine. So that actually probably better. But for the phosphorylation, for other acetylation and other things, what type of, I think it's from the EM density map currently, it's called it probably still a little bit stretch. So, but if we know from mass spec and other correlated information, we probably can, oh, okay, so that makes sense, okay. But for de novo identification, I always say it's a little challenge at this stage. So I'm not sure what other part of the question. Yeah.
Thank you.
Yeah.
Thank you so much for a fantastic and really insightful seminar. So I noticed that the structures tended to be quite flat, although you really didn't talk so much about the interactions within the fibrils. With a system like
use the mic. So
with a system like A beta 42, where you have multiple different monomer structures or dimers or whatever, do you have similar patterns sometimes of the monomer interactions within the fibrils, or what kind of diversity of interactions do you see between the monomers and the fibril?
Yeah, so since the cross beta has this common features, the neighbor layers basically form this standard hydrogen bonding between these layers, right? So that interaction is strength is very strong, and that's what holds this amyloid together. So we present the structure more or less like a flattened structure, just mostly just for the easy to view. But if you actually see the structure, there's a little bit small undulation, you know, and up and down out of the two single 2D plane, mathematical 2D plane. So yeah, so in the early publications, we actually try to show that as well. But I guess later the field has seen enough of those. The field has converted to this kind of presentation. Most of the flattened basically projected the common 2D plane to see this meandering of the backbones. But yeah, the outer plane undulation, that is for sure. Every fold has a unique, you know, outer plane undulations. Yeah.
All right. Well, apologies to Greg and Alexandra that we didn't have time to get to all the questions, and to Qing Yu online. Thank you, Wen. And I want to point out to the audience that one of the interesting things about Wen's work is that it touches so many different areas. He could have given you an hour-long seminar on virology or on computational biology. So I would encourage you to follow up reading some of Professor Jiang's papers. And thank you very much for, thank you for coming.